Today we will listen the PhD thesis defense of Othman Sofen. Othman has graduated at KFU PM, and then he decided to enroll to KAUST. He enrolled to KAUST several years ago. He has done his master's degree at KAUST, and then he continued for his PhD degree. And here is the result of several years of work. So now I am transferring this to Othman.
Okay, I have my
good luck.
Thank you. Okay, Bismillah. Assalamu alaikum. Good day, everybody. Thank you all for coming. I would like to thank the committee members, my Professor Vlad. Today I'm going to present my PhD work about novel data mining methods for virtual screening of biological active chemical compounds. So in today's presentation, I'll start giving you a brief background about the problem, and then we will visit 3 different solutions addressing different perspectives when we apply data mining to this problem of virtual screening. So in particular, we will start with Dragon wrapper feature selection, or DWFS, for handling the feature selection problem. And then we'll have Dragon active learning minority oversampling technique, or DRAMOTE, to handle the class imbalance problem. Finally, we will go with DRABAL, or Dragon Bayesian active learning for multi-label classification. So drug discovery is one of the very sophisticated processes. It takes on average 13.5 years to develop a single medicine, and around 800 million to 1.8 billion dollars are spent on that single medicine. It takes long time, starting from initial phases where we have the large set of chemical compounds, and then to later stages where there is a filtering step and you can have a better selection than the initial one, until we get to the clinical trials, which take quite time. As you see in the graph, it can take around 7 years to go for clinical trial phase 1, phase 2, or phase 3. Finally, there should be an authority like the FDA drug authority to approve that drug. Now, unless we can make this process much cheaper, faster, we cannot talk about what's promoted nowadays as personalized medicine. So in the future, we will have personalized medicine, hopefully, which means that instead of having a single medicine for a whole group of people, we'll have a medicine based on the molecular profile. Now, some of the advantages would be that, as you see in the front view, we would have many medicines. This is part of the medicines that I have at home. So we maybe can avoid this number of medicines if we are really targeting patients on their profile, and maybe we have more efficient treatment. So there are different techniques from the computational perspective that deals with this problem. Starting with bioinformatics, which deals with building insights from the biological data, chemoinformatics, building insights from the chemical type of data, pharmacophores about optimizing the supramolecular interaction between a chemical compound and target, docking studies, or protein modeling. So in my PhD, I will be focusing chemoinformatics, because giving a particular biological target, we'll be trying to find a good candidate compound that will interact with this biological target and a protein, for example. So one of the standard approaches to do this is to do 3D docking, where we get the 3D structure of the protein and 3D structure of the chemical compound, and we try to link them together in a way which is similar to a key and the lock. You are trying to find a good key for that specific lock. So there are certain issues in 3D docking. First of all, it requires the 3D structure of a protein and the 3D structure as well of the chemical compound, and that is not available for a wide range of chemicals or proteins. And at the end, it reduces to some optimization problem, which itself would be facing an issue of false positives. So I'll be doing data mining to avoid many of those problems. So data mining is flexible, actually, in terms of the type of data that we can handle. Not necessarily we have to have 3D structure. We can have any types of data. And once we train the model, we can do fast screening. And we will have accessibility to many of the existing state-of-the-art methods, because at the end, we would like to really minimize the false positives, which would mean we are minimizing an actual experimental validation in the lab, hoping that we save the costs or the money spent. So I will start with this question: how to do a faster virtual screening? And here I'll be introducing DWFS or Dragon Wrapper Feature Selection. So before I carry over, we have here a framework that I propose to handle data mining for virtual screening. So if I want to do data or apply data mining for virtual screening, we need to start with data collection, where we need to have some data. And once we have that data, we need to prepare a data in a way that is suitable to many of the existing mathematical models or machine learning. Of course, we need to have some optimization steps. At the end of the day, we can, once we have that nice model, we can do virtual screening. So I'm mentioning this a couple of times or a few times, the virtual screening term, what it means. It is a computational technique itself, which means I'll be searching a set of chemical compounds and looking at their activities, if they will bind or not, for example, with the biological target that we analyze. At the end, we have to still do some kind of results validation to confirm on our findings. So the type of data that we are dealing with is called high-throughput screening assays, biological assays, or bioassays. This is an actual photo from one of the laboratories where they will be doing an experiment. We will have a robotic arms that will take an actual plates of chemical compounds and we'll take them and inject them on some plates of the cells or or proteins, and then they maybe will have an incubation period. After that, they will get the reads, store them in the database. So this is the kind of actual experiment that they do to test the activity of a number of compounds on the biological target. Of course, once we have this data, it can be stored in the shape of a report or data set that we can have, for example, in a database. Now, one of the in fact, it is the biggest public repository at the PubChem bioassay database, where reports would be stored there, and then an investigator can come, check those existing reports, build the second assay based on the previous knowledge. So this is the type of data that we'll be using. Let me give you an actual example for that report from PubChem bioassay. So a bioassay would have a title that would explain something about the nature of the interaction. So in this case, we're talking about agonists of the thyroid stimulating hormone receptor. We look here, we find that the actual protein target is the thyroid stimulating hormone receptor, or TSHR. And then we'll have the description, the protocol that they did the test in. Now, interestingly, we have all the list in that. This is an actual lab experiment that they did. So they have tested the activities of 73,000 almost compounds, and only 1,800 of them were active. So they will report for us the actual compounds that were found to be active or inactive in this single experiment. So when we're talking about specific interaction, one target protein, for example, a set of reported and tested compounds, this is what we call a bioassay. And this is, of course, a rich type of data that we can exploit and build some model or machine learning method. So we have introduced the data collection part. Now, this is type of data that we have. We need to prepare it in a way that would suit our modeling. So we start with data preparation, and we need to define some way to represent certain variables or numbers that would reflect on these chemical compounds. And once we have this, we can do something like feature selection to optimize the performance, and maybe other further steps in doing the optimization. Then we have the virtual screening, because we have a good model. We can apply it now. We can take it actually and test it, and eventually the results validation. So now I will go to this step where we'll talk more about feature generation and feature selection. So giving a chemical compound, it is the input that I need to generate some numbers for or some variables for and explain it so that I can apply some of the methods. So for example, there is a set of keys that's called max keys, defined by experts, that may relate to some medical or medicinal chemistry definition. So it may relate to some treatments or therapeutics effects or so. There are what's called chemical descriptors, for example, the molecular weight. So given a formula, we can maybe get the molecular weight and some other numbers. Topological fingerprints is one type of features. PubChem 881 dictionary fingerprint features is one of the widely used in these type of studies. So to explain further, let's have this example, which is one of the chemical formulas here. We can directly take this chemical formula and, as I said, compute the molecular weight, and this is one variable. So other than dealing with the string, I'm now dealing with the variable and some specific number. This string can be represented in what's called the smile representation, and it is kind of simplified molecular representation. It's a 2D representation that we can use. In fact, we have many chemoinformatics existing softwares or toolkits that we can use to generate a set of numbers for these chemical compounds. And we can see that we can have a graph that would represent this kind of smile. And then we can have a predefined set of features. It's a matter of matching. So if this is part of this bigger graph, then we say this is 1 or 0. And it is a different way again to generate those features. So we generated different types of features. At the end of the day, we had almost 3,000 features. Of course, we assumed that we might have redundancy there or irrelevance features to our problem. So we need to check and go further with a more enhanced way to select a good subset of features, and that's where we went with feature selection. So feature selection is a typical step that we do in machine learning to choose of the big set of the initial set of features, a subset of features that would optimize, for example, the performance. So it can optimize the prediction performance. Once I have a minimal set of those features, I can do my screening faster. And if there is a good chance that I know which features are really contributing to my problem or chemical interaction, I'll be able to understand more about the underlying biological scenario and explanation. So here we introduced our framework, which is called DWFS. So basically we'd have a classifier, and this is well known in data mining as wrapper feature selection, where we have a classifier and there is an optimization function that we want to optimize. We want to minimize the error rate. At the same time, we would like to minimize the number of features, maybe giving some weight parameter alpha. There will be some search procedure. Remember now I'm talking about feature selection, so it's inclusion or exclusion. It's a combinatorial search problem. I can use one of the global search method like genetic algorithm or the parallel version of it. All in all, this would be the wrapper approach. Wrapper approach for feature selection is well known to optimize the performance, enhance the performance of classifiers. It is criticized for being slow. Now we thought, since we have parallel genetic algorithm, we have nice resources at KAUST, okay, let's use the parallel genetic algorithm, just optimize everything. It turned out not to be the case because it is still a combinatorial problem. The space is really huge and there is a tendency to overfit if you optimize beyond certain number of iterations. So we came up with this step. It's just a simple filtering method. So before I go to the wrapper approach, I start here. And giving the features themselves, I can use some statistics to exclude some of those features. For example, I can use the standard deviation. Remember, I'm talking about active and inactive classes. And if single variable is having a very small, let's say negligible standard deviation, it means that this whole variable is giving me useless information. So I can just exclude it. And based on that, we were able actually to exclude a set of features. And in a way, we are enhancing the search space, and then we go with the wrapper approach. And this would be the hybrid model that we have, or hybrid framework for feature selection. So let me show you here the effect of feature selection on the problem of virtual screening. Now, this is an ideal scenario. It is not an actual one. What I'm showing you here, I went to one of the existing high-throughput screening assays. We took the top 50 actually reported compound to be active, and the top 50 and the other 50 which are found to be inactive. And we represent those with some features. Now, one of the standard uses is that, in fact, if you go to PubChem, because you are having a compound that you want to query for the certain bioassay if it's active or not, and then we can use some similarity measure or distance measure. Usually they would be using a measure like DRAMOTE coefficient, for example. So what we would like to have is to group actually all of the active together and the inactive together. This is the ideal scenario that we have. Now let me take you to the actual scenario when we will have an actual feature generated. And let me say here, though DRAMOTE distance is one of the main things used, we are using here Euclidean distance because we're trying, in DRAMOTE we have to use binary features to compute the Jaccard kind of similarity. So here we're using Euclidean distance because we have continuous values in these types of features. So here we have the 1 and 2D feature. This is one group of types or types of features that we can generate for a single compound. And as you see, if I'm going to use this setup where I get the compound and just to query the most similar compound based on this type of features, there's a huge mixing. You cannot say if my most similar compound is just active, I will be active. This is the point of this figure. So there is a large mixing between the red and blue boxes. And remember again, this would be the ideal case where we have all the blue together and all the red together. We go further and we test this. We show this experiment with the PubChem widely used fingerprints and with our selected features. It is still the same. So in all cases, you will have this huge mixing. It's worth to note, however, that our selected features give the minimum number of mixing compared to the other cases. So now let's go further after we saw the effect of feature selection and virtual screening and compare actually our features to PubChem fingerprints because they are really the widely used types of features in the domain of high-throughput screening, mining high-throughput screening assays. So this is an interesting experiment that we do on that single bioassay where we have 1,000 of compounds. So the y-axis would be the testing geometric mean on 20% portion of the data. As for the steps, these different steps represent different training conditions for using an SVM. So let me explain. First of all, this is the size of our features in this experiment, and this is the size of PubChem fingerprints. Okay, so remember the problem is highly imbalanced, so we have many of the cases being negative, and some of them would be positive. Now what we do, we train an SVM, and once we train an SVM, we know we can access this information. We know what are the set of negative support vectors, and then we can exclude them. When we exclude them, we are enhancing the balance between the both classes, and so we reduce the imbalance. So we go and we continue until we achieve some good balance and we see the training. This whole experiment is actually taking us to a kind of test where we're testing the performance of these features under different balancing effects of the data set and giving different kernels. In particular, if we look at the polynomial kernel where we run the SVM with the polynomial kernel, the triangle specifically, our features were way better than the ones using PubChem fingerprints. If we change the kernel, actually this performance of the PubChem features jumps a lot, but then for the first initial steps, we are achieving better results. Now, at the later stages, we get to have a similar performance. In fact, this is the more challenging part because here where we have the actual imbalance, and this is the nature of high-throughput screening assays that we are dealing with. So giving this analysis, we say, of course, so our features are better, so we can continue the other analysis using these features that we came up with after feature selection. We took our framework, we will go and we run it on, we do thorough analysis on different data sets, and we compare it with many of the state-of-the-art existing feature selection tools. Weka, for example, for wrapper feature selection, FST3 toolbox, different variants, MRMR, joint mutual information, and we compare with all of these. So this is the performance. So this plot is giving the comparison of these different methods for a different data set. And the y-axis here is measuring the geometric mean to the size ratio, which means that I want to have this as higher as possible. I want to have performance high, and the number of features, the size here reflects the size of features as minimum as possible. So for KNN classifier in 6 out of 9 data sets, our method outperformed the others. For naive-based classifier, we outperformed the others in 4 out of 9 data sets. So far in this part, we introduced DWFS, or Dragon Wrapper Feature Selection. So this is an online tool that can be accessed. It's in a framework way where users can choose different types of objective functions and combine them with different filtering techniques to do the optimization. And I showed you actually the effect on virtual screening and how we performed on other biomedical data sets, and I performed in some cases 5 other state-of-the-art methods. Now let me move on now with another interesting question, because I'm talking about faster and cheaper virtual screening. So how to do a cheaper virtual screening? And here I'll be introducing Dragon Active Learning Minority Oversampling technique. So let's visit back this bioassay that I showed you a snapshot for earlier, this report that we had. And see here that 73,000 compounds were tested. Only 1,800 cases were found to be positive or active. Now, if I'm dealing with a typical binary classifier, I'm having this huge imbalance between the both classes. The classifier would be mostly biased toward the majority class, which is inactive in this case. And of course, we don't want to get into this situation because we'll be doing a high misclassification error on the testing set. Nowadays, in the laboratories, they are able to screen around 100,000 compounds per experiment. And only 1% is the hit rate of these tested compounds found to be active and interacting. This is an intrinsic problem in these types of high-throughput screening assay that we need to deal with when we're sorting out a data mining solution for virtual screening. So let me explain further. Let's assume now that these black circles are the negative majority class and these blue crosses are the positive minority class samples. So let's assume that we are going to query this point, so we don't know the actual label of this point. We will query it based on a classifier like KNN1, so the first nearest neighbor. In that case, if we check the closest point to it, it would be black, and I would use this point to help me to label this point. So it would be black, but it was actually a blue cross sign. So in that case, we are misclassifying a point. We need to handle this problem. We need to emphasize and enrich the space of the positive classes, but we need to keep something critical in our minds. When we're doing that, we need to keep false positives under control. Because when we are enriching that space, there is a chance that we end up actually misclassifying positive samples. One of the state-of-the-art solutions to handle this problem is called SMOTE, or synthetic minority oversampling technique. Again, here we have the majority samples and red and black circles, and then we have the red stars. Now, the method will do the following. It will pick up one of the minority samples, check one of the random nearest neighbors, pick up one of them randomly, and then do some linear interpolation between them and generate a new sample, which we call a synthetic sample. So we are enriching the space. We are adding more samples here in this region of the space, trying to enhance the representation of this minority class. Now, if you look at this method, it's actually ignorant of any feedback from the classifier. So we developed this method and proposed DRAMOTE. In DRAMOTE, we do things in two steps. First of all, we quantify the importance of these specific points. In other words, we would like to see if this point is like in a danger zone or more in a safe zone. So this point is very close to the majority class points, so there is a high chance to misclassify it. But at the same time, it has other irrelevant neighbors from the same class, call them mates, for example, that they can give it support. So I really want, because there is a high chance that I misclassify this, and I really want to give it good support. So in that way, we can quantify the importance of these points based on the feedback that we get from the classifier and the training phase. Now, once we are able to quantify these points, the importance of them, we can choose the direction where we can generate the synthetic samples. So for example, let me go back to this point. I want to generate synthetic samples, but I will not pick up any more a random point. I'll pick up the point on the safest zone direction so that I can have a good generation of the synthetic samples. And this was a very good way to keep false positives under control and enhance the precision that we have. If we go and we compare with SMOTE, the method I just explained, MWMOTE, a more recently developed version that showed was shown to even outperform SMOTE. So what we see here is that for these two data sets, we improved on precision. In fact, for all of the 9 data sets that we had in the study, we improved in the precision. Of course, we need to check as well the sensitivity. So although we sacrifice a bit here, we enhance precision much more. And this is what we care about more in this typical setup of virtual screening, because we're minimizing false positives, meaning that we minimize the actual experiments that we will do. And the same case here, this would be the results if we use a summary metric like F.5 score. We wanted actually to explore other sampling solutions, like for example, random undersampling, where we go to the majority class and we remove a set of points there. So we explored randomized sampling, granular SVM randomized sampling. These methods would be really enhancing the sensitivity, but sacrificing precision. So at the end, when we're applying actual solution, we need to keep this in mind. If we really want to improve precision or sensitivity, or maybe we combine both. So now I addressed for you a solution where a way to generate features, and then we did feature selection, and I introduced a way to handle this class imbalance effect. So now we can go with this step of virtual screening, where I can bring an actual drugs, for example, a set of these drugs that were approved by the FDA, and then see if there is new kind of outcome based on these data that were tested in the laboratory, but the result were not checked against some of those drugs. So once we do this virtual screening for these compounds and we search for some drugs that will interact in a specific setup that were not known before, we maybe go further and check other ways to do the validation like 3D docking or literature or text mining. So let me talk about the 3D docking setup here that we did. So we had, of course, protein. I'll show you a figure later on. And we had to center a grid where we do the actual search for this 3D docking. And I followed this setup where I used a tool called KAUST to center the grid, because it's really difficult to choose where you would do the docking. And remember again, there is a major issue in 3D docking where we end up with many false positives. And then we used the Autodoc Vena with the default setup and we fixed the random seed. Now we do this specific comparison between three different sets. We take the top ranked predictions for the thyroid stimulating hormone receptor. So these were experimentally validated to be top acting with this target. And then we take another part, which is, these are the predictions that we give. And the actual top experimentally validated is another set that was reported to be experimentally valid and interacting with this target. And then we just choose 10 randomly selected drugs. We want to compare ourselves to a random chosen set and to another one which is experimentally found to be active. So when we do 3D docking, we get two different metrics for evaluation. First of all, it's the energy of interaction, and the minimum it is, the better it is. So this is the experiment. This is a box plot for the three different groups: the experimental one, the predicted group that we predict, and the random one. And we achieve the best median across all. But compared to experimental, actually the experimental here did not achieve the best result. So we thought that we have to go and to check with another docking metric. In that case, we checked with the root mean square distance, which explains the distance of fit between the protein and the chemical or drug. And interestingly, the experimental was so fine with that. And again, this is the minimum it is, the better it is. And this distribution itself is nice. We are very close when we compare ourselves with the experimental one against the random sets, which is highly random in that regard. So this is the protein that the thyroid stimulating hormone receptor. It's responsible for the function of the thyroid in our human body. And when there is a misfunctioning in the thyroid, there are symptoms like vomiting, nausea, and so. Now, this is the top selected drug, which is called ondansetron. When we go and we check the literature further, we found that this drug is used to treat as well vomiting and nausea. And this is the way that we try to link from the literature, since these were not really reported to interact together. We check further the literature to see if there is some kind of validation for these predictions or findings. So in this second part, we actually developed a toolbox that we published, and we put also the data sets that we experimented with. And in that toolbox, users can try our solutions as well as the other solutions for the class imbalance problem. And we did an interesting case for drug repositioning. We did further analysis on the drugs and using the docking and the literature validation that we did. Now I come to the DRABAL or Dragon Bayesian active learning for multi-label classification. And here we are trying to address this problem, this question. Can we really enable big virtual screening where we can exploit more data other than just dealing with a single bioassay? We can deal maybe with much more. Other than dealing with a few thousands of compounds, we maybe can deal with hundreds of thousands of compounds or millions of interactions. So we revisited the whole framework and went to the step one and said, okay, if we expand this, can we achieve better results in terms of saving money spent on these experiments when it comes to validation of the findings of the predictions that we do in data mining for virtual screening? We went back to the PubChem bioassay database and we were checking the existing correlations between different data sets there or different bioassays that we have. So one clear example would be the case where we have two different bioassays, and this is the AID, which is the access ID for these two bioassays. They are two different experiments, but they are talking about the same protein target. So they have the same protein in this case. Here we have other types of data sets where they share the same protein family. The third and most interesting case for us, which we use, is where we have two different bioassays, and in fact, they don't have any commonality between the protein targets. They are totally different. But when we check the active sets between these two compounds, we find a high correlation between both of them. So we thought maybe this is interesting to exploit, because if I'm giving a prediction that correlates these two, maybe I'm really exploiting the underlying type of data and the pathway analysis of the interaction itself. So we modeled this as what's called the multi-label learning. Now, this is a typical example of multi-label object, which is a figure here, and we have different labels for this figure, for example, a lake, a trees, and mountains, maybe we have snow or white or colors or so. Now, this is different from the typical binary classification problem, because in binary classification, for each sample, we would have one assigned label. In the multi-label classification, we'll have a set or subset of assigned labels for each sample. This has been ubiquitous, actually, going for many applications for document classification, web pages, and can be applied to the chemical molecules or compounds that we have. So this is an example to illustrate more how to model this as a multi-label classification. So we have compound A that interacts with bioassay 1, 2, 4. Compound B interacts with bioassay 4. Compound C interacts with bioassay 1 and 4. Let's assume for this example that we don't have enough information about the interactions with bioassay 2. But let us assume again one more thing, that there is this rule that I can build. If bioassay 1 and whenever bioassay 1 compound is interacting in a specific data set, bioassay 1 and 4, then there is a high chance that it will be interacting in bioassay 2. So I'm interested now to know if compound C will be interacting with bioassay 2. So maybe I can exploit this correlation since I don't have sufficient information. And then we build our target matrix. So we will have compounds. Remember, compound is a kind of chemical molecule, but we represent that as a set of numbers or features. So this is a type of matrix. But then, rather than having a single vector to do the classification, now we have several vectors, and here we'll have negatives assigned labels, positives, and missing. Some cases would be missing where we don't know the outcome of that single experiment or test. So here we took some data sets. We took actually the 5 largest bioassays in the PubChem confirmatory bioassay database. They were representing 1.4 million interactions and 400,000 almost unique chemical compounds. 30% of the target matrix that I just showed you was missing. Okay, so what are the existing solutions to deal with this problem of multi-label classification? So one of the state-of-the-art solutions is called just building a binary classifier, because if I go back to the matrix, I can have a binary classifier separate for each target label. And this is, in fact, is reported to be one of the very hard to beat approaches for multi-label classification. So we can build a binary classifier for each data set, and here we will have binary relevance. Now, another way to do it, where we can exploit the correlations, is called classifier chains. So in classifier chains, we will choose the first data set, and we will be building just, again, a normal classifier for that data set. So I'll have this data set and have the target labels and try to build a model to predict these labels and assign the predictions for these samples. Now, if I go to the next step, in a classifier chains, what we will do, if you see here, we have a new data set here. However, we will go back and take the target label here and include it as an extra feature. And then we'll be predicting or modeling the hypothesis for a new target label. In that case, since we are taking this target label of the previous model as an extra feature in this second step, we are really forcing the model to try to learn or give it the chance to try to learn any existing correlation. And so on and so forth. We continue with that until we get to the last model. Now, if you see here, if you notice here, this is a white box. Why? Because here we had only 3 samples. And this is another data set which is only 4 samples. So if I take this 3 sample vector added to here, I will have an issue of missing label in that case. So this is a problem for us that we have to deal with. So one of the limitations or issues in this classifier chain approaches is actually dealing with the missing labels, which it is a characteristic of the type of data that we are dealing with. The ordering, if you realize how we are going to order these classifiers, do we do it randomly or in a specific way? Classifier chains would be doing it just randomly. And then the change size, it is forced that the second model would have 1 extra added feature and the last model would be having n-1 features. So this is kind of maybe, you can view it as a limitation or a constraint that will be limiting maybe the performance. So we proposed our method called DRABAL, or Dragon Bayesian Active Learning method. Basically, we simplify the problem, okay? So we go to these target labels or bioassays, and we really care about the ordering and the dependency between them. So we said, okay, let's just learn the Bayesian network structure of these variables. So here I would have x1, which corresponds to one of the labels, x2 and x3. I just go and learn the Bayesian network structure. And since I'm able to learn the Bayesian network structure, I know actually some nice information that I can exploit in modeling the relationships between the classifiers that I have. So, given that I've learned the Bayesian network structure of this set of variables, we can do this. Now, for the first data set, we know that which correspond to this target label, we can build a model for it. All right? So, we don't need to incorporate any information. When we go to data set corresponding to target 3, we will do the model, but we will include information from the first data set. And how we do this, because there is a trick here that we do to avoid this missing label issue. So we have a set of samples here, and we build and train an actual classifier. And then we don't care anymore about this data set. Now we will have for model 3 a new data set. I'm not going to take the previous labels as is compared to classifier chains, but I will use the model. I will give my first model this set of labels, take its predictions and as probability scores, and inject them in this feature matrix. So in this way, I'm actually quantifying those missing labels, not anymore facing an issue with them, and we continue the process till the end, and here we have our approach. We take our approach, we compare it with the classifier chains, CC with a missing label extension. Again, we have, if we are going to apply classifier chains, we need to handle those missing labels, and one of the ways to handle them is assuming them to be negative labels. In this experiment, we do this, but I will show you later on another experiment where we do something different for handling missing labels for classifier chains to have a more fair comparison. And we compare as well with binary relevance, where we use different types of classifier. In all cases, we significantly outperformed the other methods, the other state-of-the-art methods, giving different performance metrics. Now, I would like to highlight in particular this interesting part. The only difference between binary relevance with random forest and DRABAL is only 0.1%. What does it mean? We are using exactly the same classifier. In DRABAL, we have to have a classifier. Here we use random forest with the same parameter, seed, everything. The only difference comes from the fact that we changed the data sets where we put some extra features in it. And we were having five data sets, and on average, we were adding just 2 features. And the full feature space was 1064. So we're adding very few number of features. And if you quantify this, it is only 0.1% change in the data set. Even though we outperformed that other metric significantly and the others. And this indicates that even though we were really adding very few number of features, they were actually very important to the training approach that we used. This is where we fix the sensitivity for these 5 data sets based on the second best method, which is birelevance with random forest, and we see the precision, which is more interesting for us. And in 3 cases, this is the orange curve for us. The battery is off. Okay, fine. So we check the first case, the third one, and the 5th one, and we significantly outperform the other method with these data sets. And for the 4th case, we have exactly the same performance. The second one, we sacrifice a bit. We go and we do one more interesting experiment. So let's go back to the classifier chains. We can easily fill the missing labels using the same idea that we came up with for our method. So we'll use the classifier rather than assuming them as negative. That would help us actually to quantify the contribution of the Bayesian network learning step. And remember again, there is an issue of classifier chains ordering. We can order them randomly. So we run 10 different random orders. We take their average. Again, we compare with the method. In that comparison setup, we significantly outperform that setup. Okay, nice. Now we have DRABAL. We have to apply it actually. We have to take it to an actual case and see the drug repositioning that we do. So here I'll show you a short video about what's called Niemann-Pick type C. It will give you a good idea what is the effect of this disease.
Niemann-Pick C disease is a genetic brain disease. Children with Niemann-Pick C typically are perfectly normal looking at birth and develop disease only later, sometimes years later. You want to find something to stop that disease now, not something years from now, but something now.
So this is a neurodegenerative disease. It has no treatment so far. So it was a very interesting case to take into account. So we went and we did this. In fact, for 2 bioassays that we have, they were, the 2 in the middle, they were related to this Niemann-Pick type C disease. So we developed our model using DRABAL, and then we brought the drug bank database and we did the screening. Of course, we removed any common set to see the novel, exciting findings. Interestingly, we find one drug to be common between these 2 targets, the one in the middle, and you can see it highlighted by red. I think this is a virtue because we were exploiting correlation between the models, and the drug appeared to be between the 2 sets of those. And these 2 data sets, they are talking about 2 different proteins, but which are related to the Niemann-Pick type C disease. We went further, analyzing the literature about what is that drug, and to understand more about that specific disease. So this is a drug called thiamidazole. It's a drug that is originally used against a variety of nematodes, types of forms that can live in the human body or the animals. It is described as an oral hydrocarbon receptors that reduces levels of a protein called cathepsin C protein. And the overexpression of that specific protein is one of the characteristics of NPC disease. So we are examining the literature, trying to see if there is connection between this drug and the symptoms of that specific disease. One of the things that we did is going to a database that's called Stitch database. And in Stitch, you can query, give it an input of concepts, and see, and it will give you back a set of concepts with the correlation in a graph structure. So I'm sorry I'm not able to use the pointer here again, but what we have here is a set of concepts, right? So these circles represent proteins, and these kind of ellipsoids represent the chemicals. And whenever we have a blue link, it is linking 2 proteins. And we have a green link, it is linking a protein and the chemical. And if we have a red link, it's linking 2 chemicals. Now the weight or the thickness of that edge represent the enrichments of these concepts. These concepts are coming from actual publications in the literature. So they mine some of the existing literature and they try to relate them. Our query was NPC1 and RAP9A. And these are 2 proteins, the names of 2 proteins which were found in these 2 data sets. And these 2 proteins, they are related to the NPC disease. When they are overexpressed, they are found to overcome some of the symptoms or phenotypes of the NPC disease. So complementary to the findings that we found using our method that this specific sample dissolved drug is going to overexpress those, we find the following. A benzoate group here. Now this benzoate, this is thiabendazole and that's a benzoate group. They share a benzene ring. It's not a major kind of correlation, it's some kind of indication for us, but it was not part of the query. There are other interesting concepts that appear in this graph, the cholesterol, for example. If we go back and we read more about the MPC disease, it is characterized by extra accumulation of the lipids or the cholesterol in the lysosomes of the cell. So there were reports, interesting reports, that would say if we use thiamidazole with different doses, we are able to, the level of the cholesterol in the serum cholesterol level would be increasing. So let me simplify it for you. So we have this part of the cell where we have the cholesterol accumulation. It's not able to get out, and that's why we are having a disease. Normally, it should be getting out, going through the blood, transferring to two different parts. Now, this is not happening if we have MPC disease. What we found out is that maybe if we use this drug, it can be released and flow normally through the blood. Though the best setup would be to actually test it in an experimental laboratory. So in this part, I showed you a novel definition of mining high-throughput screening assays where we correlate them based on the active set of or common set of chemical compounds. And we handled certain challenges like the missing labels, ordering, and chain size of the classifier chains. So we went to the laboratories. We took the useful reports there. We had to do some data preparation where we can represent them as set of numbers or features. Following that, we handled the class imbalance problem. This is one of the major problems that characterize those high-throughput screening assays. At the end, we tried to correlate these different types of data sets in a single approach to do to enable us to correlate the targets and maybe enhance our predictions. So in all, we have Dragon wrapper feature selections published in PLOS One, DRAMOTE for handling class imbalance PLOS One, and recently DRABAL just got published in Journal of Chemoinformatics. So, because our main goal, one of our main goals was actually to enable big virtual screening. So we went and we put all of these solutions together in one single system that users can use. So what you see here, this is an interface of the system. Now, in the top there, the user can choose different types of inputs. The PubChem AID, for example, which is the PubChem bioassay ID number, and extract some useful information. The user have the choice also to upload chemicals or other types of informations like smiles and so. And then we have the Dragon wrapper feature selection features that we selected, and other different types of features that the user can select. And in fact, he can or she can use the different combinations or feature selection methods. We as well added the toolbox to handle the class balance as part of this tool. And we give the choice of the user if they want to correlate different data sets, they can upload the different bioassay IDs and we'll be correlating them. Once the user builds a machine learning model using this system, this model can be taken and actually screened against the available drugs in the market, the available approved drugs list in the market from drug bank database, for example. Then there is a chance to build a graph for visualization. And since we are giving predictions that would correlate or relate the protein name with a specific drug to be active, we can query PubMed or existing literature and retrieve an interesting set of literature to help on validating or help the user in finding some more validations about the predicted interactions. For the future, I'm proposing this. So I'm looking this at the future as we have two different directions towards the personalized medicine at the top right corner that we want to reach. If we really want to go there, we need to work on with much more data variability. At the same time, this is not enough. We need to have accessibility to more and more of experimental types of data that were validated in the laboratories that we can find them to strengthen our findings. So we go on the x-axis from a basic setup where we have chemical and protein, to further steps where we can maybe combine different types of information from the food, from the environmental chemicals, from DNA methylation, mutations, different types of molecular information, and do mining in that setup. As well, if we can access more and more types of experimentally validated results. This is a list of references that some of them that I used in this study. So there were some other projects that, of course, I worked on during my PhD. Some of them I list here in this slide. So I worked closely with Dr. Wael Ba'alawi, where we were trying to address the problem from a different perspective of graph mining. We were trying to do graph inference there, and we published that method and, in fact, filed a patent for the method of graph mining interaction lists between drugs and targets. I participated in several data mining challenges. Very recently, we participated in this very interesting challenge, the AstraZeneca-Sanger drug combination predictions, which were about combining the molecular types of information with the combinations of drugs and to see if those drugs would be giving a synergistic effect on killing the cells of the cancer or not. I was really honored to get this chance during my PhD. Through delegation of Professor Vlad and Professor Panos, I had the chance to supervise Muna Shahrani for her master thesis, Muhammad Muafa during internship project with Professor Panos, and Mustabal bin Saad right now having directed research. Reviewing for +1 applied soft computing. I've been the TA for the applications of AI and bioinformatics with Professor Vlad for 2 semesters right now. So in my PhD, I published 4 journal papers. One paper is actually, the 5th one is now under review in applied intelligence. 3 others are in preparation. So we're planning to submit the system that I introduced to Nucleic Acid Research, which will open the submissions for web servers on the next month. DAS5 Plus, another interesting work that we did with Wael Ba'alawi. We had a submission and we got the good feedback. We are going to work on that and submit it again. And one class classification for characterizations of different types of drugs, which we hopefully, we are going back and revisiting some of the interesting data sets and building the new updated ones. Hopefully we can submit that close to bioinformatics. Previously, before I just start my PhD here at KAUST, I published 2 conference papers, and we were able to file a patent with Wael again for that method. So what's the next step for me? I'm going to hopefully continue this in line with what I'm doing. I'll be dealing with more chemicals, maybe that would relate to food, environmental chemicals in the McGill-McDonald campus. And my task would be there actually to find the top list of predictions for certain cases or tests. So this is the campus, is somewhere there, and I hope that you will visit me someday. You will be finding me somewhere there, hopefully. So I'd like to take 1 or 2 minutes here, if you allow, please. First of all, I'd really like to thank Professor Vlad. Previously, Wael said that you had always our back. This is, of course, true. Tanveer said in his defense that you were like a father for us, and this is true. And what I like to say to you, actually, that you give chances for people, and you gave me chances until I succeeded. So I really appreciate this. I feel really sad to go away and not to see you, but you promised that you will visit me, and I promise that I will be visiting you. I would like also to thank Professor Panos Kalnes. We worked closely on this PhD journey. I learned a lot from you, Professor Panos. It was actually very interesting to see the different perspective from the problems that we do. Your group is working on the graph mining approaches. We were really getting nice ideas, and some of these things that we have here were based on brainstormings and many of the discussions and meetings. I would like to thank Mr. Professor Stefan Arrow. He's in the middle of the conference, and he found a good spot to join us. We are missing your smile here. Thank you, Professor Stefan. Professor Takashi, though I didn't meet you except a few times, I think your wisdom shows always in those meetings. Thank you so much. Professor Christian Shomba, thank you for coming all the way from Japan. Appreciate it a lot. and appreciate your comments, which were very relevant to us. I would like to thank Wael Ba'alawi and of course Mu'taz, Afif and Makbouba. In some parts of this research, it was really tough. This is a multidisciplinary research. I cannot just do computer science and apply it. So we had to engage biochemist Makbouba and Dr. Makbouba and Mu'taz. They were really supportive because sometimes the conflicts raises between computer science and biochemist or a chemist or a bioscientist, but things were going fine. Now, there is a big list of people, maybe I can mention some names. I have to thank Professor Shingao because he was following up with me for job applications during the last period, and he wrote me a good recommendation letter. Professor Xian Liang, she wrote me a nice recommendation letter that got me to work with the Professor of Lab. Our Dean, of course, all faculty from CBRC, Abdullah Khamis, Haytham, Arturo, Tanvir, John Hanks, and Sultan, and all CBRCs, and InfoCloud, the team of Professor Panos, Ibrahim, Razin, Zuhair, Demetrius, and all InfoClouders, and from KAUST, Ammar Mir, Mahmoud Ouda, Ahmed Shuel, the Dahwa brothers, Luqman, Yanal, Abu Talha and his family, and all community. From KFPM, the people who support me to work on some research and get me excited about biomedical data applications before I come here, Dr. Samir Arafat, Ayham Dada, and all KFPMers, mentors, Sheikh Abdurrahman Hirfi and Khalid Saeed, relatives, brothers, sisters, and Allahs, and many, many others. And if your name is not that, it's not here, it's in my heart actually. And I have to thank at the end, very dear people, I have to thank my father. And my father actually is a businessman, and he was looking always for the chance where I finish my bachelor and I go back to him. And honestly, if he did not support me, I wouldn't have been here today. Whenever I'm talking about science, he's always supported me to go and continue. Of course, my mother for her supplications and emotions. When she's satisfied, I just fly with happiness. My wife, of course, it's not a normal life, and most of the days for PhD, I take my laptops from the work, I go because I'm computer science. This is my laboratory, so just a small laptop. I take it to home, I continue working for hours. Maybe in the weekends, she wants to go to Jeddah, she can take the bus because I'm busy to work on my laptop. But she made everything looks normal. I'm really appreciative for her. And of course, for my nice kids. Last of all, I really want to thank my homeland, Syria, because what's going on there is giving me really good reasons and hopes to go back one day there, to work hard and go back one day there and contribute to the educational system based on all of these standards that I've gained. And all praise to Allah. Thank you all, and I welcome all questions.